Hello, everyone. Thank you for joining us, and sorry for the short delay. Welcome to this presentation, which is part of an ongoing GOOS webinar series about aspects of global sustained ocean observing. My name is Albert Fisher. I'm the director of the Global Ocean Observing System office here in IOC in Paris. And for the next hour, we'll start with an approximately 30-minute presentation from Laura Lorenzoni. Laura is a researcher at the University of South Florida in the USA and also has recently spent time as a program scientist at NASA. She will be speaking about Marine Ecological Time Series. After Laura's presentation, we'll conduct a question and answer session by chat or through the question box. I will moderate and select the questions verbally, and the chat window will be open during the presentation if you want to start asking clarifying questions. The session is being recorded. A link to the recording will be posted on the GOOS website, and you can find it as well on the GOOS webinar YouTube channel. Laura, I'm going to hand it over to you for your presentation.
Thank you, Albert. So it's being recorded. That means I have to be very careful about what I say. I want to thank GOOS for inviting me to give this webinar, and everybody for joining. Oh, you can see me. So I can't make any weird gestures either. That's going to be a problem for me. But no, again, thank you, Albert, for this opportunity. As Albert said, I'm going to be talking to you for about half an hour about an effort that we've been undertaking for the past roughly 4 years, which is the International Group for Marine Ecological Time Series, and what we've been doing. And the aim of the group and the aim of this presentation is to highlight ship-based biological and ecological time series and the unique information that they can provide us on marine ecosystems and how they are changing. The work that I'll be highlighting it was led by the IOC, the Intergovernmental Oceanographic Commission of UNESCO, the International Ocean Carbon Coordination Project, the IOCCP, and the Ocean Carbon and Biogeochemistry Program, OCB, here in the US. And of course, all the logos on the top of the main presentation slide are the people or the institutions that contribute to this effort, which include KLS, Pisces, ICES, and NOAA. And I am merely a sheep herder. The other people that helped me with this are Todd O'Brien from NOAA and Kirsten Eisenstein from IOC. And we had a fabulous and absolutely wonderful steering committee that did a lot of the heavy lifting. And of course, I wouldn't be even saying anything about time series if it wasn't for the time series themselves, the PIs and everybody else that goes out time after time and gets all that valuable data. All right, so why are we here? Well, to talk about time series, that's why I'm here, to highlight the importance of sustained observations through time and understand what kind of information they can give us. Most of the times, the data they provide, we seek it to answer questions, but most of the times those data generate more questions, which is what makes this such an important endeavor to sustain. And while time series, you know, one of the weaknesses of time series themselves is they provide sustained observations, but only in a very limited spatial area. So it is one spot. And so they can provide, probably, if you look at it just the one time series, they can provide information on, you know, the local, maybe the regional variability of where it's taking a measurement. But when you pool time series together, you're actually obtaining, you know, a broader footprint, and that is the value of actually, you know, grouping and pooling the time series together. And that is what IGMETS, which is the acronym, went after. And time series are part of GOOS, the Global Ocean Observing Systems. They're a very important part of it. And sometimes it's, can you still see me? Sometimes they're, I don't want to say neglected, but they're sometimes overlooked. Just like remote sensing data. A lot of times we don't necessarily think about it as part of the Global Ocean Observing Systems, but they are. And something sad that comes back to sustaining ocean observations, ever since IGMETS started in 2014, we have lost 7 long-term time series due to personnel and funding issues. excuse me, the Bay of Fundy Phytoplankton time series, it was a long time series, and it ended because the person that was actually leading that time series retired, and no one stepped up to take that place, either because there wasn't anyone prepared, or maybe they didn't have the funding to replace that position. And the Cariaco Ocean time series is another one that we sadly lost this year, and it had over 20 years of data, and that was lost because of a variety of conditions, including funding. So when we start losing these old time series, it's a hit, because once we miss that data, we're not going to get it back. And because of the cost of time series, there's been a lot of discussions on whether we should go to automated sensors in trying to replace some of these measurements that time series are doing. And that is it's a very good push, because we can take some measurements with automated instrumentation. However, as we are now, we cannot substitute a lot of the biological and ecological measurements with autonomous sensors, unfortunately. So we can try to augment, these two things go together. We can try to augment current observations with some autonomous sensors. We cannot replace the actual ship-based time series. And that is something that we need to keep in mind. It'd be like going to the doctor and just getting a CAT scan and not even going to the doctor itself, just trying to trust what your automated sensor tells you without going and looking at the ecosystem. So we would be missing a lot of information. So the fact that we need to sustain ship-based time series and sustain ocean observations as a whole is a topic that has recently been at the forefront of a lot of national and international initiatives. So it's not, what I'm telling you is probably not new to you. However, it's frustrating that we've been talking about this for decades and we're still talking about it. I am hopeful that maybe the time is right and the stars have aligned. I'm going to mention three efforts that are ongoing at this moment, which hopefully will continue to give it push and maybe move us forward. Currently, the G7 has a working group focusing on the future of oceans and seas. And one of their actions is specifically sustaining ocean observations. And they're particularly focusing on what they call augmented observatories. And that means adding biological and ecological variables to existing time series sites. So they're basically augmenting existing observatories. GOOS is working closely with the G7, and that is very important because the idea of trying to leverage existing observatories, so for example, areas of GOOS interest with these augmentations and trying to link that to G7 support is critical to move ocean observations forward. And the last thing that I'm going to mention is the US has recently released a report by the National Academies of Sciences on sustaining ocean observations. So we're talking about it. We're not there yet, but I'm hopeful that there's momentum gathering that we can move this field forward. At least try to find a way that certain key ocean observations can be sustained. Oh, and so the last point, I can't not mention the far side cartoon, which is what happens when we miss data. Well, we miss data and we actually have no idea what's happening because we're not observing. So we are foregoing important information that may be critical for us to predict, some important change in our ecosystem. So I mentioned in my previous slide, we're all geared towards the right direction, or the time is right, and we're gathering momentum. But I was in a meeting recently, and I was deeply concerned about a couple of things that I heard that made me realize that there is still a lot of disparity in our community on what is the right direction in terms of how do we sustain ocean observations, and in particular, ship-based time series, marine and biological and ecological time series. One of the first comments that I heard was, we need more data. That is a very scientific thing to say, and I very much appreciate it, but we don't need more data. I mean, we need targeted data, and we need to sustain that targeted data. And for that, GOOS has been working for already several years on what is called the essential ocean variables. Excuse me, and they can help guide efforts that we're doing in terms of sampling. So we just don't need to go out and collect, all the data you can collect. You need to be smart about it, because that also is going to impact the type of investment that you're making on your time series. And the other comment that I heard was, well, we should give a prize or token of appreciation to these folks that dedicate their lives to sustain ocean observations. That is a really sweet thought. But I would argue that rather than giving them a prize, these folks, deserve to have some sustained funding to carry out their measurements without having to struggle every funding cycle, or whether they're going to, be able to go out and take those measurements. I mean, there are other ways than just a token of appreciation that they need in order to carry out their job. And time series are not, these biological and ecological time series, they're not just taking a bunch of measurements. They're measuring our ecosystem. They're establishing baselines, which is information that is critical for any decision making that any government is going to make. And so, born out of this sort of frustration of the lack of recognition and the lack of support that time series are having at a global level, that's how IGMETS was born. So IGMETS wanted to create something which ended up being a report and a bunch of other tools that you're going to see throughout the presentation, to provide a means to highlight all these ecological time series and provide some of the information easily digested of what you can gather when you pull all of these resources together. And hopefully the idea is that makes it all the way up to decision makers, and that they can see the value of these sustained ocean observations. So let's get into it. So what is IGMETS? And ahead of time, I apologize. The next probably 3 or 4 slides have a lot of text. But since this presentation is going to be made available later, I'd rather keep the context in there. So if anyone needs to go back to it, it's easy. So IGMETS, as I mentioned earlier, started in about 2014 as a means to assess global and regional ocean change through ship-based biogeochemical time series. And the idea from the start was to combine multiple time series, which would enable, rather than just a local or regional assessment, push it up to a more broader scale. IGMETS wasn't born, just one single day. It was sort of the product or the baby of a bunch of other efforts that came before it. So it includes GLOBEC, ICES working groups, IOCCP and OCB workshops. And so for the first time, we brought together, through the IGMETS effort, more than 340 ship-based time series worldwide to look at how the world had changed over the past 30 years. And yeah, as I mentioned, the goal is to highlight the importance of this continued sampling carried out by existing ship-based marine time series. We did not get into autonomous time series, and those have their own value. I don't want anyone to feel that they're not valued because of, and you're going to see we had our own issues with ship-based time series and methods. Because autonomous assets are sort of a different beast, we stuck with ship-based time series, and the idea was to highlight them to begin with. So it's not like we're ignoring the automated ocean observations. We just didn't include them in this effort. And so this is the vision that IGMETS had, and I'm going to briefly read it. So it was to encourage the use of already established common metadata, analyze time series data at a global level, increase the visibility of these groups, allow the possibility of web-based tools to analyze some of the time series data, and provide a strong base for future predictions that could advise policy. And the image that you're seeing here, all the little stars are the time series that actually participated in IGMETS, and those are overlaid over a satellite chlorophyll image composite. So most of you are familiar with the figure that I put here from Deke Otto. When you analyze one single time series, you get at the scales that the orange square refers to, so you can identify, establish your local baseline, identify temporal patterns, understand local processes, but then once you group all the time series together, you're actually getting a bigger footprint, and you can actually look at teleconnections, you can look at larger climatic influences on regions, and so it provides a whole new dimension of analysis that you can do on ecology, which is something that is very hard to do. So who is IGMETS? Currently, as I mentioned earlier, myself, Todd O'Brien, and Kristen Eisenstein are still sort of the leading folks that sit on IGMETS. Luis Valdez was absolutely critical to getting IGMETS started. He was at IOC when this started. Now he's at the Spanish Institute of Oceanography, and I'm sure he's very happy there. We had an absolutely fabulous steering committee. The names are listed there, and they all been absolutely wonderful. And of course, we had all the time series investigators and participants. So how has IGMETS worked? I'll tell you one thing, it was a really long process. I mentioned it started in 2014, but we started thinking about the whole IGMETS idea probably a couple years earlier than that. So gathering the data, which is the basis of what IGMETS sought to do, was also very time-consuming. It took us over 2 years to get all of the data from the different time series that ended up participating in this effort. We actively reached out to all the time series that we knew, and we saw it at every meeting, at everybody that we talked to. We tried to find whether they knew of a time series somewhere. And it was a real discovery effort, and it was a lot of fun to see all of a sudden time series emerging out of the woodwork and going like, hey, yeah, I've been taking measurements here for the past 6 years. And so that was really exciting, because you found people that were as dedicated as we were, and as excited to be part of this, to be able to provide their little grain of sand to this type of effort. So as I mentioned, most time series were excited to be part of this, but data itself is it's a sensitive topic. We were very conscious of these sensitivities, and data policies vary. They vary between countries, they vary within countries, they vary within institutes, and so we came to find this out as we were gathering all the data for the report, and being sensitive to this. How we got around having time series participate and not feel alienated because they couldn't contribute openly their data is that we did not publish or release any of the raw data that the time series provided, but rather what we released are the products of those data. And so this was actually advantageous for the PIs as well, because they were free to utilize all of the IGMETS products for their own publications. So the other thing we tried to do for PIs was that we told them, send us what you have in whatever format you have, and we'll take care of it. And we were fortunate enough to have Todd O'Brien from NOAA, who was like absolute genius and wizard with the data. So he took all the data, he formatted it, and he generated all the products. So this was a minimum effort from the PI side. And the PIs were involved in every step of the way. So we asked them to check once the products were generated, does this make sense? Does this not make sense? Once the write-ups were done, we had them read it. Is this accurate according to what you know about the system? Because in the end, the PIs are the ones that actually know how their region works and how their time series, how they take the measurements. They've seen things that maybe we don't see necessarily from one variable to the other, and they can actually make those connections. So having them involved in every step of the way was critical. And we also used satellite remote sensing, specifically sea surface temperature and chlorophyll. And satellite data was critical in order to provide context to all the different time series measurements. So even though you're pulling all the time series together, you're getting a regional and a global perspective, but the remote sensing tie it all together. So satellite data was critical. So what type of analysis did we do? Well, first off, we divided the world into regions, and this is specifically for the report that we came out. that we came up with. So because that made the analysis a lot easier, considering the different dynamics of each ocean region. The second thing, well, you know, when you have data from different sources or from different time series, you hope you know, the methods are consistent. You hope the reporting is consistent. But the truth is, there's, you know, you get apples, you get oranges, you get a lot of stuff. So which is to be expected. So our goal was to be able to compare, you know, apples to apples and oranges to oranges, and then be able to, you know, put them side by side and say, okay, so what is the fruit doing, or where are the different variables doing? So the first thing that we came up with was we derived just unitless trends, and then we used the non-parametric Mann-Kendall test to test for significance of these trends, and how to analyze all these trends and all these data. Well, what we did was that we divided the data into different time windows, and the table that you have in the screen is the different time windows that we used. So we had 6 time windows, starting from 30 years all the way to 5 years, and we'll come back to the time windows later. And the IGMETS analysis, as you see on the 30-year time windows, went from 1983, and we stopped at 2012 because it just wasn't practical when we started in 2014. A lot of the time series data, the ecological data especially, and the biological data, it takes a long time to be processed. So when you have zooplankton and phytoplankton, it is just not realistic to seek data that is like really up to date. So we felt 2012 was a reasonable year when we started this in 2014. So by now, you know, those data are already 5 years old, and so it is something to think about. And so at the end, we generated, with all of these data, including the satellite, we generated trend maps. And this is sort of what the trend maps look like. What you're seeing here is the background is the satellite data, and so this is, in particular, this is a 10-year trend map of satellite in the background, and the legend is at for the satellite, it's at the very bottom, so the background fields, and then you have the rates of change. And then all the symbols represent the different time series that we're actually measuring sea surface temperature and their specific trends. And this is what the trend maps look like for chlorophyll. And again, it's the same thing. The background is the satellite chlorophyll, and the different symbols are the in situ chlorophyll. So what else did IGMETS do in terms of analyses? Another product that we came up with was what we call the brief overviews of dynamic ecosystem, or the Bode plots. The truth behind the Bode plots is that Antonio Bode, which was one of our chapter authors for the North Atlantic, he came up with the idea, and so we had to find an acronym to name the plots after him. And so, being good scientists, we managed. So the Bode plots are actually really useful because they are a very quick way to visualize the relative amounts of positive and negative trends, or the correlations, depending on which plot it is, within a given time window and across a set of in situ variables. So for example, in this case, at the very top, you have the Bode plots. This is for the North Pacific. That illustrates within a 10-year time window how many time series had a positive or negative trend in a certain variable. So, for example, 15 time series had negative trends in temperature, and 8 had positive. 17 had, declines in zooplankton, while 5 had, increases in zooplankton. And then the plot in the middle, all in red, it shows the variable correlation, so the in situ variable correlation with sea surface temperature, in this case in the North Atlantic. So for example, 90 time series were negatively correlated with sea surface temperature, where 93 were positively correlated. And the same thing goes for the bottom one. So you had 126 time series that were correlated with satellite chlorophyll, which is fortunate. And 61 that were not, which is interesting, because not always the satellite and the in situ measurements agreed, and they didn't agree for very good reasons, and we went into detail about that in the report. So lastly, IGMETS generated what we call a spatial temporal frequency table for temperature and chlorophyll. And what this table does is that it provides sort of a percentage of what area of a certain basin increased or decreased significantly or not in terms of temperature and chlorophyll. So what you're seeing here on the screen is actually for the North Atlantic region. So you had that, over the past 10 years, about 50% of the Atlantic has warmed significantly, and another 50 hasn't. So it varies within the time chunk that you choose, which is an extremely important thing. You have to remember that where you start your time series and when you end your time series, it's going to give you a certain trend. So it's something to keep in mind, and we discussed that in the report a little bit. And you can play with these time windows. So in terms of products, IGMETS first came out about halfway through our effort. We came out with what we call the discovery map, and it is downloadable. It's a PDF. It's really pretty. Kristen Eisenstein led the design and completion of this. And it illustrates what you see on the screen. It's only the front part, and all the yellow dots are the time series that participated in the IGMETS effort. On the back side, you have actual details on all of those time series. So it's a very nice thing to have. We then came out with a report which was released earlier this year, I think it was July or August of this year. And the last thing that we have produced are the web tools. One is the meta database, which includes all of the information on the time series that participated. And then the Explorer, and what I showed you before about the trend maps, that is part of the Explorer. So it's a lot of fun to play with that. And you can access all of this in the links that I'm providing. So the report is 297 pages. And I would make a terrible disservice to all the authors and people that participated if I attempted to summarize the report in the next 10 minutes. So what I'm going to do next is I'm going to provide you very briefly some bits of some notable results that I thought were cool when I was putting this presentation together. But I would invite you all to read it. It's a great, right before going to bed, nothing better than to do but to read the report. It's really makes you have good dreams. So we like to think it's a useful report, and if you do end up reading it and you have comments, we welcome any type of feedback. This is the first time we've done something similar, and so any feedback that we can get from the community is most welcome. So in terms of notable results, I think the upper picture, the upper panel is sea surface temperature in different time windows. So you have 5, 10, 15, and then all the way to 30 at the bottom right. And all the bottom panels are pretty striking because you see the amount of red that it's in there. So one of the notable results was, of course, there is a generalized warming trend over the past 30 years that was seen, both with sea surface temperature from ship-based and satellite data. And of course, this is in agreement with all the IPCC reports. However, there were regional differences which were very interesting, and depending on the time window that you selected, you could see, for example, El Niño's influence on sea surface temperature. So it was interesting to see all the different time spans and see how the large-scale climatic variability was impacting some of the in-situ variables. And one other thing to note is we see this warming, and IGMETS ended in 2012, as I've mentioned. So we didn't include 2015, 16, or 17, which are some of the warmest years on record. So it's going to be interesting to see, once we expand this time series, what the temperature is going to look like. The middle panel is chlorophyll. So of course, satellite chlorophyll has only been available for the past roughly 17 years. So we had 15 years of, we had just 3 time windows for chlorophyll. And the trends for chlorophyll were a lot more variable. So there wasn't a sustained trend that we could say, like in sea surface temperature. It varied by region significantly. And I think some of the most valuable things that we came out with in the report was that seeing as time progressed and seeing the different time windows, we could see in relating, basic hydrographic variables with the ecological and biological variables, we could see that there were actually shifts in the biology and the biogeochemical cycling. And these didn't just happen at a single time series. Once we put the time series together, we saw, there's this shift occurring, for example, in the Baltic Sea or in the entire Mediterranean region, or the South Atlantic has really changed in terms of biology and ecology because of different reasons. Everything. Oh yeah, and the last panel at the very bottom is actually a correlation between sea surface temperature and chlorophyll. So we also generated correlation maps, which were useful and very surprising. So another thing in terms of notable results was the Indian Ocean. First and foremost, it was striking to see how the Indian Ocean has changed. And over the past 30 years, we noted about a 92% increase in its area. So 92% of the Indian Ocean significantly increased in temperature, in sea surface temperature. And there was a paucity of in situ measurements. I think we had one time series in the Gulf of Aqaba, and we had about 3 or 4 time series on the west coast of Australia. And that's it. So with the type of changes that the Indian Ocean has been experiencing, and that includes also declining in surface chlorophyll, it's obvious that there have to be some severe ecological and biological consequences and changes, but there are no time series measuring those changes, which is of concern. India has about 3 time series at least off the coast of India. They, however, were not at liberty of distributing their data. So it was very unfortunate, because I think those would have been extremely valuable to understanding changes in the ecology of the Indian Ocean. So another important thing that we noted, something that most of us know, was, of course, Arctic Ocean waters have warmed over the past 30 years. About 79% of the Arctic has warmed. Chlorophyll has increased slightly, but it's been very patchy. And the problem with the Arctic is that it is very complex. And again, just like in the Indian Ocean, you will see that there are very few time series that are measuring any ecological, biological variable in the Arctic. So it made the analyses very difficult. And of course, all the changes that are happening in important regions like the Arctic are anticipated to have severe impacts to Arctic resources. And again, the image that you see here, you can see that also in the Explorer. So it's an easy thing to play with. By now, and hopefully you caught this when I showed the first map of the background chlorophyll with all the little stars of the ship-based time series that participated. I hope you caught this early on, and you'd be like, huh, I see an awful lot of stars, adjacent to the continents, and I see a whole lot of nothing in the rest of the ocean, which is true. Most time series, most ecological and biological time series are located along the coasts of, the continents, it just makes it for easy logistical access. However, that leaves a large gap in the ocean in terms of measurements of ecology and biology. And the other thing that you hopefully have noted is that I don't think any more stars fit in the continental margins of Europe and North America, but there's a striking lack of time series in other continental margins around the world, and so that makes these type of analysis very hard, because we're kind of getting a skewed vision, and as much as we need to understand how ecology and biology is changing globally, we're just not getting that view. So obviously we need a more globally distributed network of time series to understand how the ocean is changing and how our resources are changing. Part of it is capacity building, but it's just a very complex problem, and I'm not going to pretend that we can solve it in a couple of days. So I mentioned that I was going to come back to the time series, to the time windows. So we had, you remember we had 6 time windows, 5, 10, 50, etc. And when we looked at the analysis, we tried to look, how's the world changed, how's the ocean changed in 30 years, 15 years, 10 years. And we came with the 5-year time window. We were like scratching our heads and thinking whether the 5-year time window actually makes any sense. And the fact is that it is kind of short and it doesn't have a lot of statistical significance because of the shortness of the time. However, if you look at this figure of number of time series and the length of time, you'll see that most of the time series are babies. They're young, they're on the 5-year. And so we had, when we looked at different variables, we did have a lot of data on the 10-year and 5-year end and a lot of very scarce data on the older side, which is a problem because the older time series are the ones that are getting shut down because they can't get support. And those are the ones that are most valuable because those are the ones that have the history, that have the baseline, and those that we can actually use to predict changes. I'm not saying we don't need to sustain the younger time series, but it's a matter of trying to sustain also the old ones. So where are we now? The report, as I mentioned, was released earlier this year in July 2017. We have the different products that we've already gone over, and I encourage all of you to go and take a look at them if you can. And in terms of what the future of IGMETS is, we were selected last year as another IOC working group, so efforts to highlight time series will continue. But we don't want to do the report just over again and add, I don't know, 10 years of data. We want to give it a different light. And this is when I mentioned that I think if you have any feedback or if you have any comments on how we can make this better or different for the next installment, it is most welcome. We're looking to maybe try to pull together different data, so maybe higher trophic levels. It is a bit more complicated, but it might be something useful. And if we put things in a perspective that can be of strategic importance to different countries, maybe they will appreciate more the efforts that each time series does. So we welcome feedback on future directions and how to make this type of product better. And most of the time series, they're very excited to keep this effort going. So this is my last slide, and this is not a concluding remark because this is a continuation. This is not over by far. We still have a lot of work ahead of us. It is becoming increasingly obvious that we need to frame the importance of ocean observation and sustaining ocean observations within frameworks of importance to each country. So we have to give it a different spin. I think the days of doing science just because we love science are long over, and we have to think of applications and other ways that we can sort of sell the mission of sustaining ocean observations. It is also important that we coordinate as an international community. As I mentioned at the beginning, this is not my thoughts. I think these are the thoughts of hundreds of people that have the same concern, and I give credit to all those people that are trying to find ways to sustain ocean observations. But it is better if we're coordinated rather than if different efforts try to do the same exact thing at the same exact time, because then we're also competing for attention and resources. So coordination amongst the international community is critical. And we're at a good time. I think all the things that are happening make this the right time to at least try to push this forward a little bit. And as I mentioned also, IGMETS will return, and we welcome any community feedback to make this better. And with that, I'm happy to take any questions if you have any. Thank you.
Thanks a lot, Laura. That was a really great talk and really big effort of a lot of people that you're representing and presenting to us all. So I am turning over to the audience for questions. We have some specific questions already, and I'll start with a couple general questions of my own, maybe, to warm things up. You mentioned quite a few times the sensitivity around the sharing of data and the data policies and how that varied between countries. Did this exercise of pulling all the data together and building some scientific value out of the sharing of that data, did that break through in any country? Did that, do you have a sense that people are more open to sharing their data in the future?
That is a very good question, Albert. I think they do. I think it's important to build a trust of any participating, whether it be time series, whether it be a single investigator. I think it's important to build a trust that they can share their data and nothing is going to happen. It is important to understand that a lot of the countries that cannot, or the institutions, we found, it was very interesting, we found sometimes that there were people not sharing data within institutions. So the physical side would not share data with the biological side. And then, so it was a very interesting dynamic of which, you know, we can't judge why they do things or what they don't. So we had to go with that. And a lot of the countries that don't share data openly, they rely on their data to push maybe their tenure or their salary. So it is a very sensitive topic. Once they realize that maybe nothing bad is going to happen if they share their data and someone is not going to run off with their data, because that's happened more times than I can count. Even with data that's shared, you don't even know they're using it and you don't get credit. I think it does build a sense of, well, maybe I can share their data, maybe I can participate, but it's a very slow process. So it is a very good question. I think it's a matter of patience and being respectful.
Thanks, Lara. And then another sort of GOOS-oriented question. I mean, obviously these time series were all measuring different things, or many different ecological parameters, but were there, can you say sort of broadly which set of essential ocean variables were being measured across all of the time series we looked at?
Yeah, so for the, we focused on, we focused on biology and ecology, obviously, and biogeochemistry. So they were mentioning a whole suite of physical essential ocean variables. The ones that we pulled in were temperature and salinity, because those were the ones that we were interested in. In terms of biogeochemistry, they were measuring oxygen, nutrients, some carbon parameters, but there were so few the ones who were measuring carbon parameters. We did not deal with organic matter at all, dissolved or particulate. I think those are the ones that we pulled in: the nutrients, oxygen, and some of the carbon parameters. And for ecology, we focused on phytoplankton and zooplankton. And those are the ones that most time series are measuring. If you try to go to, you know, different other ecological and biological EOVs, the data is even sparser.
Okay. And
but the report has all the list of variables that are measured are in the report.
Okay. Another question about, this kind of a general question. I asked you a question about whether people were more willing to share their data after the exercise. Are people more interested in doing things to certain standards or doing things in a standardized way to make the intercomparison between the time series easier?
I don't think they really care about intercomparability up to a certain. Let me take a step back. Intercomparability may not necessarily be the priority of their list. I think what they care mostly about, and we saw this in the workshop that we did back in 2012. IOCCP and OCP sponsored an intercomparability workshop for time series. They were mostly concerned about the quality of their data, and once you follow a certain standard and you get, you know, units that are comparable, your quality is generally good. So their main concern was the quality of the data, and then being able to compare those data with other time series. The intercomparability comes after you make connections with other time series, and a lot of the time series are actually focused on, am I doing it right? They hadn't even gone to the step of, can I call such and such, and can we understand what's changing? So that is the next step. And time series, ship-based time series, they take so much effort that most people are just so focused on collecting their data that they're not thinking about the broader picture. So hopefully this serves as that catalysis. And the one thing that we did find a lot of resistance in some of the time series in terms of, are you doing it right? Are you making data that can be compared with other time series is, I've been doing this way for 10 years and it's easy. So changing that, because not necessarily it's correct, was met with some resistance. But younger generations are open to change, and so as personnel turns over, that is a more of a possibility. So it's a matter of how you sort of introduce the concept.
All right, let me jump into some of the questions from the audience. You have mentioned the report a number of times, the heavy report, and so we have a question from Jennifer Skinner, who says, as you said, funding is one of the main constraints in sustained ocean observing. Are there any plans to communicate the findings from your report to those who control the purse strings? Policymakers are unlikely to read all 290 pages, so I'm interested how this will be communicated more widely.
I had a problem. No, I'm joking. I had to read it. No, it is an excellent question. Thank you. Absolutely. It unfortunately did not materialize before the report was released, but there is, so the first page of the report is an executive summary, and that contains, all the highlights and all the important stuff. And then with Kirsten Eisenstein through IOC, it is anticipated that we're going to do sort of a summary for policymakers. And this is something typical that IOC does with all of their reports, because nobody's going to read 300 pages of report. In terms of nobody in the government is going to read it. So absolutely, there is a plan to do the IGMETS for policymakers and sort of highlight the important things. In terms of funding, that is a trickier question. And the report that I mentioned earlier in my presentation about the national academies sort of was providing alternatives on how to find funding. So charities, well, not charities, private entities that provide funding, like the XPRIZE, that sort of thing, were mentioned in terms of areas where you can find funding for ocean observations. Well, unfortunately, most of those venues, they don't like to sustain funding. They mostly like to fund something like a snapshot. So I don't know how reliable that may be as a venue, as an avenue for alternate funding for time series. It is a struggle, and it is something that we need to keep thinking about. It is a very good question.
Okay. Let me ask a question about, you've talked about the scientific interest in these time series, but the need linked to the funding question to link it to more practical outputs, to perhaps linking it to policy or management of ecosystems. Is there examples from all of these time series you looked at where that's been successfully put into play, where the information from the time series has been used in a policy or management context?
Canada, I think it's a prime example of how they ingest all of their. They have a long history of zooplankton and phytoplankton time series, and so all of that feeds into Fisheries Canada, and they use all that information from the time series to understand how to manage their fisheries stock. Coffee is a classic example of how a time series was born, trying to understand the fisheries problem, and so it's still going strong after several decades. And so those two examples that I gave, unfortunately, are in the Northern Hemisphere. It is a bit more of a struggle in the Southern Hemisphere, but most of or in other developing countries, but most of the time series that emerge in developing countries, they do have to make a statement on how they are important for the country itself. And so that they're trying. We've seen success stories about that. We've seen not success stories about it. So it is possible.
And you've looked at large-scale trends by combining lots of different time series. Is there other types of complementary data? You also showed some remote sensing data, but is there other types of complementary sustained ocean observing data that can help you situate a single time series in a broader context and perhaps increase its impact for those things like of policy and management?
Absolutely. And I mentioned we stayed away from autonomous assets, but all the bioargo that have been deployed, those contain at least the basic oxygen. And we had very few time series that had actually oxygen measurements. And oxygen, or the lack of oxygen, or how oxygen is going down in the ocean, it's a genuine concern. So if we were to pull in all the different, or at least some of the autonomous assets to provide more of a complementary picture of at least the biogeochemistry in the ocean, I think it would enhance our report. And we may try to do that in the next installment. We were just very scared about the volume of data we would have to tackle. Todd was, he was absolutely fantastic, but he was like, oh my gosh, I don't think anyway. So yes, absolutely. There are other data sets that we can pull in to provide a more complementary and more holistic picture of how the ocean is changing and how that affects resources. So I think also the next step is to look at higher trophic levels, though it is very challenging.
So just to follow on that, let me ask this specific question from Marcus Stucker. I didn't catch whether you included or not the automated measurements, and if not, whether this was because it was hard to automate biological and ecological variable measurements, or because the automated measurements cannot be trusted, or some other reason. And you started to answer that question, but maybe you can answer in more detail.
Thank you. So, and I apologize if I wasn't clear. So we did not include the automated measurements. We stayed away from them mostly because just pulling the ship-based time series was such a big endeavor, and we only had Todd to work with it, and he did a fabulous job. So we stayed away from automated assets. I think you have to look at automated data carefully. I think the data is good, and at least for the bioarchive, they have a very rigorous QA/QC process. So it is a matter of making sure you understand what kind of data you're ingesting. And I think there's really good data out there that we could definitely use. We were just scared of the volume of data that we would have to tackle with. In all truth, the idea of this IGMETS installment was to highlight biogeochemical and ecological and biological time series. So we felt we wanted to focus on that, at least in the first installment.
Let me read a comment from Frank Muller-Carger. He once mentioned that OBIS, the Ocean Biogeographic Information System, GOOS, and MBON, which is a marine biodiversity observing network of GeoBON, so part of the group on Earth observations, they're working together, are supportive of the IGMETS effort, encourage engaging in these networks for capacity building, adding biology to observing systems using standards and shared data via OBIS, and IOC and GEO are behind such collaborations. Do you want to add anything to that comment?
No. I'm
okay.
No. Thank you for
and then let me get back to Johannes Carstensen is asking a question which links your effort back into some other parts of GOOS. So great talk, Laura. Thank you. How do you see a link to the Open Ocean Time Series under the Ocean Sites Network? And maybe the more coastal time series organized in DBCP, which are mostly moored time series?
That is a great question. Thank you for raising that. And I'm actually looking forward. I know the Ocean Sites a while back had the MOIN, the minimalistic sort of how do you need to augment your open ocean observing system so that you can get a more biological and ecological view of the ocean. So I am excited about that idea. We can absolutely link to all those open ocean observing sites by including those data are automated. So as I mentioned, we sort of steered away from autonomous data in this report, but I think if we want to really capture a sense of how the global ocean is changing, we're going to have to utilize every asset that we have. And I think we wouldn't have been able to appreciate that until now that we've done this effort. And I think actually incorporating all those the oceanside sites and all the coastal sites may give us an appreciation of what we're actually missing and how can we strategically place biogeochemical sensors and co-place sensors to provide us with the maximum return, because we can't measure, we don't have any reliable way of measuring like real biology in an automated manner. And we have only some ideas of, well, you know, if we augment here and there, well, we can get this. I think it isn't unless we do this at a global effort and we pull in all the data that's available that we can actually understand, gee, we have a gap here and our resources might be better spent this way to understand ecology. So that is a very interesting way to move forward, and I think we need to give that more thought.
So talking about gaps, you showed on the map certainly lots of places in the coastal ocean and the open ocean that have gaps in these time series, at least in your analysis. You also showed the density around the US and Europe of the time series. Is there any duplication? Are there time series that could be stopped because they're showing exactly the same thing?
No, I cannot believe you asked me that question. No, I think each time series has emerged because it has had a specific mission. I think after time series have had some time to run and take their measurements, you sort of grow. And I know this having worked at time series, at a time series site. So you sort of grow. So you stop thinking about your own little, pond, because there are other things that are actually affecting your own little pond that actually come from outside. So that's how you naturally begin to expand and try to understand what is happening around you. And I think a high density of time series is good. I don't think you should overlay one time series next to the other, and that would be absolutely duplication of effort. I don't think we've come across any of that in our effort. And the stars do look one on top of the other because they're kind of big. But there is a really good spatial coverage that can actually allow you, at least, for example, take the west coast, the east coast of the U.S., which has, it is like just carpeted with stars, but they are distant enough that you can really have like a continuous picture of how the ecology and biology and biogeochemistry of the east coast is changing, because you have this great enough time series that take all these measurements and have taken them for such a long time. So you can, one measurement complements the other. So we have not come across that type of duplication of efforts. And each time series has their own mission. And so killing one because there's one down the road that is taking another set of measurements for a totally different objective wouldn't really be fair.
All right, I'm just warming you up for the question that the funders are sure to ask. We have a question from Hassan Mustafid, who wants to know whether the data from IGMETS exist in OBIS, whether it's, I don't think all of it does, but does some of the data show up in OBIS?
Thank you. That's a very good question. So each time series, we reached out to the time series individually because that was our goal. We try to really, find who was taking time series measurements and try to get them on board of this effort. What each PI does with their own data, that is their own business. We encourage them to share the data, obviously, and we try to put it in a repository because that's where it is safe, and that is where, if your computer disappears, or you get hit by lightning, or you get run over by a bus, and everything is on your hard drive, those data are as good as lost, which is a huge loss because you just can't recover it. So we're very much encouraging time series PIs to at least, not necessarily OBIS if they don't fit in OBIS, but if they have a national data repository where they send their data, or if they want to send it to an international data repository, there's IOD, there's others, there's Pangea. So there's plenty of places where data can be put and it can be stored in a safe place. However, that we can only encourage the PIs, and those the PIs have to take it up. How we managed to get all these time series on board was ensuring then that we would not distribute or release their raw data. We would only release the products because they have their own data policies, which we have to respect.
Let me ask you a follow-up question from Marcus Stocker, who thanks you for answering his question. He hadn't heard of IGMETS before you presented your work, and he finds it quite interesting in the light of the many observatories that are being built. In Europe, many of them are part of environmental research infrastructures, and not just in the marine domain, but covering other domains such as the terrestrial domain. And they're largely doing automated measurements. I think the question is, do you have any estimations of the actual value of such data?
As in monetary value?
Yeah, monetary value. What kind of decisions are you helping to make, and what kind of economic value do these data have?
Okay, so in terms of the value of the data, I'm just gonna quote is it the MasterCard or the Visa commercial? It's priceless, and it literally is priceless. These data are priceless. In terms of economic value or impact value, which is I think how I'd like to think about it better. We're hoping that decision makers pick up this report and can actually find, and again it comes back to having the sort of executive summary for policymakers, having a tool that highlights not just temperatures increasing, chlorophyll is decreasing. It's actually the value of IGMETS that it is tying it to ecology, and so you're knowing that in a certain area diatoms have gone down, and diatoms are actually the food for the fish that you actually fish. So if you're seeing that an entire region, say in Europe, say in South America, is the phytoplankton or the zooplankton stocks are decreasing, that actually is going to have impacts on the fisheries. And so what we're hoping that the policymakers see is the value of, gee, there are these resources that are already taking the measurements for us, and that we can tie our decreasing fishery stock with these environmental variables, and thus understand why we are not getting as much fish and how do we better manage these fish stocks. And so we're hoping they can reach out to their own time series in their own countries and understand the type of measurements that they're taking and provide some support. That is what I'm hoping some of the impacts of the IGMETS will be. Maybe I'm being too optimistic, I don't know, but it's what we're striving for.
No, it's great to hear the optimism. So, Laura, we've run out of time.
Yes.
I want to thank you again for your talk. It was a great talk, and we had a lot of questions. I wasn't able to get to all of them,
Thank you.
I'm sure Laura will be willing to answer these questions.
Absolutely. I mean, and you know what? I just realized I did not put my email in my intro slide. I'll make sure I correct that before I send it to Forrest. So if anyone has any questions on IGMETS or anything, and our contacts are in the IGMETS website. So you know, just feel free to just shoot me an email. I'd be happy to answer any questions. And again, thank you, Albert, for inviting me for this webinar. Thank you, everybody, for either staying, you know, at past 5 in the afternoon or early on the West Coast or any other place. I very much appreciate it. Thank you.
All right, thank you, Laura, and thank you to the audience. If you enjoyed this, please join the GOOS mailing list by going to our website to get information about future webinars. Thanks very much. Goodbye,
Thank
everyone.
you. I know how to do this. I can do this.